Hello, Wayne County Community College students. This is Dr. Morrison. Today's lecture is on Chapter 2, the chemistry of biology. Let's get started with this. Matter. Anything that has mass and occupies space. That would be like gases, liquids, and solids. Gases, say we were just using water for an example. You steam water. You boil water, the steam would be a gas. The actual water that comes out of the faucet would be the liquid. And if you freeze that water, it would be known as a solid. Those are the three states of matter. A while ago, somebody tried to introduce plasma, but that didn't quite work and didn't quite make it into these states of matter. Organization of matter. Matter is composed of individual building blocks known as atoms. An atom is the smallest indivisible unit of matter, or the smallest indivisible unit of an element. It cannot be subdivided without. This S with a line over it means without, and I use that as shorthand sometimes, so if you see an S with a line over it, will mean without losing its properties. So an atom cannot be subdivided without losing its properties. Well, as you know, we have learned how to split an atom, particularly the hydrogen atom, and the splitting of that atom, we did divide it, in other words, and the properties became explosive. So we use that to drop to that explosive property of splitting the hydrogen atom. We use that to kill other people, called a hydrogen bomb, right? So you can do something with the atom, you can split it, you can divide it, but it will lose its properties. Atomic structure. The atom is composed of 3 subatomic particles. These 3 subatomic particles will include protons. Protons are positively charged particles, and they're located in the atomic core. The atomic core, we're going to call it the nucleus. That's the very part that contributes to the atomic mass, or the mass of the atom. So protons are positively charged. You remember P with P, protons, and P for positive. Protons are positively charged particles. Neutrons are neutral. N with N, neutrons are neutral atomic particles. They're located in the atomic mass. So in other words, protons make up the atomic mass, but neutrons are also included in that mass. So protons and neutrons make up the nucleus, okay? They're located in the atomic mass and contributes to the atomic. They're located in the atomic mass and contributes to the atomic mass, okay? Electrons. Electrons are negatively charged atomic particles, and they revolve around the core. They revolve around the nucleus. And the electrons have little to no mass, right? So you got protons and neutrons work together to make the core, or the nucleus, and then these electrons kind of rotate or revolve around this nucleus, or the core. We're going to talk about mass in a bit. So we have the rest of this is subatomic particles. So subatomic particles, the protons, the neutrons, and the electrons come together in specific and varied combinations to form unique types of atoms called elements. So if you've ever seen the periodic chart, and there's one in your textbook, it shows carbon, it's in chapter 2. It shows carbon, nitrogen, potassium, fluoride, all of the elements known to man. These 3 subatomic particles come together to form all the elements known to us in the world. Gold, silver, lead, iron, etc. Atomic number. The atomic number is the number of protons. Remember, protons are positively charged particles. The atomic number is the number of protons in an atom. In an electrically neutral atom, one that would have, of course, the number of protons, which are positively charged, will be equal to the number of electrons. So that atom would be considered a neutral atom, because the same number of protons you have that are positive will cancel out the same number of electrons, which are negative. Atomic mass, the sum of the protons and the neutrons. That is the mass of the atom, and we talked about that when we talked about it being in the core or making up the nucleus of the atom. Let's look at this C, which represents carbon. Please note that this top number is 6 for carbon, and that is the atomic number, What is in the atomic number, remember? Yes, protons and neutrons make up the atomic number. If someone were to say, oh, on the periodic chart, they took away all the elements and just left the atomic number there, 6 or 8 or 7, whatever atomic number is, but in this particular case is 6 for carbon, I would still know, or the scientists could still know exactly what element that is, because the atomic number for carbon is always the same, universally. It is 6. It has that number of protons and that number of neutrons, and that makes the carbon. So if this were some other number, like I think 8, an atomic number for 8, that would be oxygen. So I would know that atomic number 6 is specific for carbon. Atomic number 8, and I hope I have that right, let me just check real quick, would be for oxygen. Atomic number 7, that's a different atomic number, that would be for nitrogen. Okay, so nitrogen would be 7, oxygen would be 8, carbon is 6. Now the difference is the atomic mass. The atomic mass is the sum of these protons and neutrons, the sum of them, the atomic mass. Well, this number can change, the atomic mass can change. Sometimes you have a carbon, but in this case, a stable carbon would be the atomic mass would be 12, okay? When they change though, when the atomic mass changes, but it's still the same atomic number, so it's still going to be carbon, but the mass is going to change because the number of neutrons are going to change. That is called an isotope, right? So isotopes are atoms with the same atomic number. Remember, atomic number 6 will always stand for carbon, but different mass number. This number could change. This could be carbon number 14, and we do use carbon number 14 for historical dating of things. We look at fossils and we can put some carbon 14 on it, and it will show the half-lives and show us the dates of certain fossils like dinosaurs or trees that have fallen centuries ago, and that's how we use it, right? So you will have a different number of neutrons when you have an isotope. Now, remember, isotopes as well, some isotopes have an unstable nuclei, and they spontaneously release energy. And that energy will be in the form of radiation, right? So you can have radioactive isotopes, and they can play an important role in research and medicine. You can use them to sterilize and sterilization processes for sterilizing your food, or using it to use it as a certain kind of dye, like an iodine. You can use that in medicine to put into your veins and look at your thyroid gland. There are different ways to use these isotopes. But sometimes radiation could be a big problem. If you ever heard of Chernobyl, there was a nuclear waste explosion that happened, and it's in I think someplace near Russia or in Russia, and now it's just a vast wasteland because of all this radioactivity that's still leaching out from that waste. So you can things can be used for good or bad. You can use the isotopes, unstable nuclei that gives off radiation for good, or you can use it for bad, right? So that's isotope, all right? Another thing I wanted to mention was some people get since we're talking about mass, some people get mass and weight mixed up. So mass, in order to have a mass, that's something that takes up space, right? So, and mass is measured in kilograms. So your mass is not dependent upon gravity. No matter where you are in the world, you will still be the same mass. However, weight is measured in newtons, and weight is dependent upon gravity. In other words, I could take this same, well, just take me for example. I could take myself, and here on Earth, I need gravity in order to have a weight, okay? But I still have I still have a mass, right? The mass is not going to change. So now take this mass up to the moon. There's a much less gravitational force on the moon. So I still have this mass that I went to the moon, but the weight is going to be different. It's going to be much lighter. So because the gravitational pull is much lighter. Is much less. That force is less. So a heavier or much stronger gravitational force is going to make the weight seem heavier, way heavier, but the mass will stay the same. A gravitational pull or force that is much lighter, even though it's the same mass, will cause a lighter weight. You get me? All right, so that's what I try to tell my doctor when I go. I'm like, really, I don't weigh this because on the moon, I weigh a lot less. So you'll remember that way. All right, let's go on and look at electron configuration. So here we are back, we were talking about isotopes last time. So isotopes are differ in the number of neutrons. Therefore, they will have a different mass number. Remember isotopes, right? Let's go on with electron configuration. Electron configuration, these are electrons, that's with the E with a little line, kind of over to the right of the E. Electrons are arranged around the atomic core. Remember, that's the nucleus. Electrons are arranged around that nucleus, and they're in orbits, or something we call shells. The first electron shell closest to the atomic nucleus can accept a maximum of 2. So the electron shell that's closest to the nucleus can only take 2 electrons, the maximum of 2, which all others will take a maximum of 8 electrons. All the other shells or orbits can take up to a number 8, which we call octet, right? So here, let's look at the atomic number of carbon, which we know carbon, the atomic number is 6, right? So let's look at a orbit of electrons with the carbon number of 6. So here is the 6 protons in the nucleus, right? So that we'll make that the nucleus. And here's one of the shells or orbits. Now the shell or orbit that's closest to the nucleus, and I'll put that as 6 protons because we see that's carbon 6, it will take 2 electrons. And so we'll just make those little dots as electrons. And electrons, of course, orbit or circle the nucleus. Now, since we have to get up to 6 and that's only 2, we need another shell or another orbit. Now this shell, the next shell can accept up to 8, and the next shell up to 8, next shell up to 8. All the others can accept up to 8 electrons. It's called the octet. So we have 2 here, 3, 4, 5, 6. Now that fills the requirement for all of the electrons, because you have 1, 2, of course the electrons in the orbit nearest the nucleus, which is 6 protons here, can only accept 2, but the other orbit can accept up to 8, but you only need 4 because that is 1, 2, 3, 4, 5, 6. Okay, let's look at the oxygen. Oxygen has an atomic number of 8, so we'll put that as the nucleus, 8 protons. Circle that, that's the nucleus. So the electrons closest to the nucleus, we know can only have 2. All right, so we need 6 more because that'll make 8. Make that 1, 2, 3, 4, 5, 6, 7, 8. Okay, so now we have, let me try to make it a little bigger so you can see them. We have 1, 2, 3, 4, 5, 6, 7, 8. And that will fill the orbits for, or the electron shells for oxygen. Let's look at valency. Valency, that's the number of electrons in the outermost shell of an atom. So the valency, here's the most outer shell here, and we know that that's 6 electrons in that valence. Here in the outermost valence or outermost shell here, that valence would be 4 electrons. So that's the outermost shell of an atom, and that's the valency. Let's look at atomic interactions. Atomic interactions are 2 or more atoms that interact to form chemical bonds. So let's talk about how atoms interact to form chemical bonds, right? Let's look at the ionic bonds. Ionic bonds are due to the loss or gain of electrons. This loss or gain of electrons is by interacting with the atoms. One donates or loses electrons, and the other accepts or gains electrons. Here is sodium and chloride, okay? Sodium chloride. Sodium with atomic number of 11, and chloride, Cl, with atomic number of 17. So let's see what that is going to look like. I gotta find enough space to make this, so I'm gonna just go up here, and we're gonna put 11 protons, so that we'll know that is the nucleus. And let's go on and put 2 electrons around it, first shell, 2. We have to get up to 11. That's just so I'll remember 11, 12, 13, 14, 15. That's 4, right? 4, 5, 6. How many can the second shell hold? How many electrons can the second shell hold? And the third or fourth or fifth? Of course, yes. Maximum 8. So I'll put 8 here. So that's all it can hold. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? So that's not 11. So we need another orbit, another shell, and then we'll put that one there. So now it's 11. But this, so this is for sodium. That's sodium, Na. Now let's do chlorine. Oh, that's 17. So that's going to be quite a bit. Let's make this 17 protons here. Put the electrons here. That's 2. 1, 2, 3, 4, 5, 6, 7, 8. You know that's 10 total, right? Needs another shell. 10, 11, 12, 13, 14, 15, 16. And then I'm going to make this one, just so you can see it here. How many is this on this? 1, 2, 3, 4, 5, 6. And this is going to be 7. Uh-oh. I don't know if you can see that's kind of red. So 1, 2, 3, 4, 5, 6, 7. This shell would be a lot more stable if it had 8 in it. It's fine. It doesn't have to have 8, but it's an octet rule that it can hold 8. This shell would be a lot more stable if it had 8 in it, but it only has 1. So let me see if I can't make that red. The point is that now, just so you'll know, this is chloride, Cl. This electron from the sodium will come over to the chloride to kind of stabilize now this outer shell and make it an octet. So this one will come over. Let's just put it here. Okay. Now that's 8, and now they can share a bond, and that will make this stable. This one, the sodium that lost an electron, became more positive, because remember, electrons are negative. It became more positive, and since it lost an electron, that's called oxidized. So sodium was oxidized. Chloride that was able to accept that electron and make this nice outer shell a full octet, it gained an electron. So it made this one more negative. So let me put this as sodium. It became more positive, so that's positive. Chloride accepted the electron, it became more negative, and that's called, since it accepted the electron, that's called reduced. I know, it gained an electron, but it's called reduced. Totally opposite of what you would think. This is called a redox reaction. Oxidation reduction reaction. The loss of an electron makes the element more positive, and that's oxidized. The gain of an electron makes the element more negative, and that is going to be reduced. And this is sodium chloride, which is known as table salt. This is table salt. And this is why it looks like this, because of this interaction here. Sodium chloride. Oh, and remember, that's a positive and that's a negative. Okay, so those are ionic bonds. Those are ions. Salts are ions, like electrolytes, etc. Okay? Let's look at covalent bonds. Covalent bonds are due to an equal sharing of electrons between atoms. This is common in organic compounds. For example, just remember it's common in organic compounds. They're bonds due to equal sharing of electrons between atoms. I'm trying to see what example I have here. I don't really have an example, but that's just the way it looks when you have, just say, a carbon to, say, an oxygen. That will be a covalent bond. We'll just leave it at that. Hydrogen bonds are bonds due to weak attractive forces between nearby atoms or molecules, such as in water, right? So here you have H2O. H2O, we know that that's water. So you just have the oxygen here and just very light, not really harsh, strong bonds like a covalent bond. So I kind of like made those like little dots. So that you can see there is attraction enough to hold the oxygen and the hydrogens, hold the hydrogens to the oxygen, but they're very light forces. Ionic hydrogen bonds are very weak, so that's why I kind of just put it like that. But it's strong enough to keep the properties of water. Remember, water is a universal solvent. In other words, it is the liquid that can, that solutes can be dissolved in, right? Or suspended in, whatever. But water is a universal solvent, okay? So that brings me to talking about polarity. Polarity. Let's see if I can squeeze it in here. Polarity. Polarity has to do with the distribution of electrical charge. Polarity has to do with the distribution of electrical charge. So we can have polar and we can have non-polar, right? What kind of electrical, how's the distribution of these electrical charges gonna be? So when you think of polar, you'll think of something that dissolves in water. Dissolves in water. What's polar that dissolves in water? Like salt, sugar, something like that. I don't know why I had a brain freeze. But salt, sugar, things like that can dissolve in water. So they would be considered polar. Non-polar are things that does not dissolve in water. And that would be similar to like lipids, right? Things like that with fatty things, fat and oils do not dissolve in water. Those would be non-polar. So my daughter, I always used to get these like mixed up, which is which. So I'm just going to share you something that my daughter, when she was in 3rd grade, told me how to remember this, and it has worked clearly every time. So when trying to remember something that's polar, think of a polar bear. Polar bears like water. So these are things that can be dissolved in water, or they like water, right? Non-polar, then that would just be the opposite. So this will give you at least an introduction into the types of bonds that we're going to be discussing: ionic bonds, covalent bonds, and hydrogen bonds, and the differences between them. Know about oxidation, reduction, molecules, when you're, and when something is oxidized, and then when something is reduced, right? So oxidation includes any reaction that causes an atom to lose electrons, and of course, because all redox reactions occur in pairs, it follows that reduction is the result of a difference from gaining these electrons, okay? So I want you to remember that. Now I want to introduce something called pH. pH. pH is the measure of the concentration. So this little bracket here, that means the concentration of a hydrogen ion, or the hydrogen ion concentration in a medium on a scale from 0 to 14. This is all over the world. We use the same scale everywhere, 0 to 14, to check the pH of coffee, for example, the pH of tomato juice, of ammonia or bleach, or your blood, for example. Our physiological blood pH is 7.35 to 7.45, so within that range. And you want to keep it within that range because if it gets a little higher, the 7.45, like 7.47, 7.50, that's going to make your blood too alkaline or too basic. But on the other hand, if your blood gets below 7.35, say it's 7.30, 7.28, that's going to mean that your blood is too acidic, right? So keep those things in mind. The pH definition is the negative logarithm of the concentration of hydrogen ion, a hydrogen ion. So just to give you what we're measuring when we're looking at pH, and then what the pH is. So here I have a scale from 0 to 14, because we know that the pH is measured on a scale from 0 to 14 all over the world. And we have 7 in the middle here, and 7 is neutral. In other words, it has just as many hydrogen atoms as it does hydroxyl atoms. So it's a balance, it's neutral. They cancel each other out. 0 to 6, all these pH 0, pH 1, a pH of 2, pH of 3, pH of 6, that means that that substance will be considered acidic. Anything higher than 8, going to 14, a pH of 8, a pH of 10, a pH of 12 or 14, those items or substances will be considered basic or alkaline, okay? That is the way we measure pH. There was something else I was going to say about this, but just so that you will get a general idea about pH, I do want you to know what is considered basic and what is considered acidic. So a low pH is acidic. A higher pH, higher than 7, is going to be basic, also known as alkaline. Our blood pH, when we take our blood from the artery, for example, our blood pH, physiological pH, is 7.35 to 7.45, right? And that's important, especially if you have or know someone in your family that has breathing problems, maybe COPD or which would be like an emphysema or asthma. They'll take that person's blood pH and see, oh, is his blood pH getting too low? If it is, then they know that they have to correct that. You have to keep yourself and your blood pH in balance, right? All right, that is pH. Okay, we're back, and we're going to talk about, before I get started, let me go back over pH. It's just something I remember what I wanted to say. So, because really when we measure pH, we're really looking at the concentration of hydrogen ions, right? So if a solution, just say, if I have a beaker here, and I have a beaker here, and it has a lot of hydrogens in it. Just put a lot of hydrogens. These are hydrogen ions, so hydrogens, all these hydrogens. A beaker with all these hydrogens in it. This solution, the more hydrogens you have, so the more hydrogens, that makes the solution more acidic, okay? An increase in hydrogen ions makes a solution more acidic. On the other hand, here I have a beaker, and it has a lot fewer hydrogens. So let's put that plus there. put that plus there. Hydrogen in the beaker will make it more basic, also known as alkaline, okay? So the more hydrogen ions there are, the more acidic a solution is. The fewer hydrogen ions there are is basic. Also, something that makes it basic too is if you have an increase in hydroxyl groups, that will also make something basic, okay? But like I said, when we check for the pH of solution, we are really looking at the hydrogen ion concentration. So that's one thing I wanted to clarify. Let's look at this. Reactants and substrate. Yield, that's what this line means. It means it yields products. So A plus B, which would be the reactant, also known as the substrate, yields the product AB. Now just say 1 plus 2, which is the substrate or the reactant, yields 3. Sodium plus chloride, these should have their little things there, okay. These are ions, if you recall. Sodium plus chloride yields sodium chloride, which we know is table salt. So when you read the reaction, read it from left to right. Some can go reverse, but since I'm getting ready to talk about enzymes, enzymes usually don't go reverse, usually just make a reaction go in one direction and one direction only. Let's see what a solution is. A solution is a mixture of one or more substances called solutes. It's called solutes, uniformly dispersed in a dissolving medium called a solvent. Do you guys remember what we said was the universal solvent? Yes, the universal solvent is water. Now say we wanted to dissolve something in that solvent. Say it's salt. We're going to put salt in there. The salt now becomes something we call solutes. The particles are the solutes. The solvent is the liquid. That whole thing is called the solution. The solvent, the solute is the solution. Okay? That is how I want you to remember it. It's particularly important when you check your urine. Okay, so let me just draw another beaker here. So they have you urinate or you pee in a cup, then they check it for, well, you can always check it to see how hydrated you are, or you can, of course, check it to, you can check urine for a lot of things, not just a drug test, but a lot of other things. But anyway, when it's really, really cloudy, or really, not cloudy, but really, really yellow and has a strong smell to it, that means it has a lot of solutes in it, a lot of particles, and less solvent, less water. That can be an indication of dehydration, right? So you never have to go, oh, am I dehydrated? All you have to do is look, once you urinate, you pee in the toilet, look at it. If it's kind of clear to just a real light yellow, you're hydrated enough. If it is a darker yellow, very amber, then you know that you need to drink more water because you have too many particles, too much solute in that solution. So I'll just, the reason I brought that up is because I was going to say something about specific gravity is a way that we check for hydration, but I'll leave that for another time. But a lot of particles, when you urinate, pee in a cup, whatever, a lot of particles, which are solutes, are going to indicate you need to drink more water. If I had urine that looks like this, I would say, that person's very hydrated. They have very few solutes, very few particles. So that's how I want you to kind of relate these things that we're going over chemistry. Let's look at enzymes. Enzymes speed up the rate of a biological reaction. Enzymes are substrate specific. So enzymes are like a key in a lock or a hand in a glove. They're specific. So this, let's just draw an enzyme, or we'll draw the substrate like that. And then we'll draw the enzyme that has to fit this substrate. And their enzymes are very specific. So, well, I drew it better. It would have to fit in here very specifically. And we don't care what this part looks like. But this connection here, that must be specific and perfect. Like a key going into a lock. Enzymes are very specific. Also, usually enzymes end in ACE. The suffix will be ASE. Not always, but usually. That's how you can tell there's an enzyme. Sometimes we have some, especially when we have enzymes for proteins. They don't end in ACE, but like I said, it's most of the time. The next thing is hydrophilic. Hydro meaning water, philic means love. So a hydrophilic substance will be water-loving. Of course, if it's water-loving, you know that's polar, right? Polar molecules like salt, or polar molecules that can dissolve in water will be like salt or sugar. Hydrophobic, hydro meaning water, phobic meaning the fear of. Water-hating, I think I got water-hating, I guess to go with water-loving. But water-hating is really water-fearing. These are non-polar molecules such as lipids, we call it like fats, butter, oils, things like that, and benzene. Amphipathic. Amphipathic is another way of saying it's not water-loving or water-hating. So let's look at it. It's neither water-loving or water-hating. Amphipathic substances have both hydrophilic and hydrophobic properties, such as the phospholipids in the cell membrane. And each one of our cells is covered by a phospholipid surrounding it, a phospholipid bilayer, which we'll talk about in a minute when we get to fats. Right now, let me introduce carbohydrates. Okay? Carbohydrates, polysaccharides. Polysaccharides means, poly means many, saccharides means sugar. So this is a polysaccharide would be many sugars. Something like a starch. A complex starch would be, just say a potato. That is a polysaccharide, right? We can eat that potato, but we can't really start to digest it yet. So something in our saliva, an enzyme known as amylase. Please note that it ends in ASE, so that means that helps us to know that it is a enzyme. So amylase has to break down the polysaccharide, which means many, poly meaning many sugars. Let me see if do I need to write that? I'll just leave it. So polysaccharides, poly meaning many sugars, uses amylase, the enzyme, to break it down to something that's a little more digestible, called a disaccharide. Now disaccharides are still not able to be digested just yet. So there are specific enzymes that disaccharides have to use. So di meaning two. This is two sugars. These enzymes, so let me write down the sugars first. So what are the two sugars? So polysaccharide, we said it would be like a starch, like a potato, right? Here, disaccharide, this is two sugars. So we have sucrose, we have maltose, and I'm just gonna and let's see, what is the other one for some reason? Yeah, lactose, the most famous one of all, right? These are sugars, they're disaccharides. So sucrose is just regular table sugar. And then, of course, lactose, right? Okay, so let me say something. So here are the disaccharides. We have to use enzymes to break down these disaccharides, these sugars, so we can get it to a monosaccharide. Why is that so important? Because monosaccharides are the only things that we can digest. We can't digest a polysaccharide, so we use amylase to break it down to a disaccharide. We cannot digest a disaccharide, so we're going to use these enzymes to break down these disaccharides, these two sugars here. Like sucrose is glucose and fructose, those two sugars. Maltose is glucose and glucose, and lactose is galactose and glucose. So let's use these specific enzymes to break it down. What enzyme do you think you would use to break down sucrose? The enzyme would be sucrase. What enzyme do you think we would use to break down maltose? Good. The enzyme will be maltase. Lastly, what enzyme do you think we will use to break down lactose? Good. That enzyme will be lactase. Lactase is an enzyme that many adults lack. They do not have it. That makes them lactose intolerant. And you will know if you're lactose intolerant because you'll have gas and bloating, and anytime you eat a dairy product, ice cream or cheese or drink milk, anything like that, you will end up having some bloating, a lot of gas or flatulence, etc. So a lot of adults end up not having that enzyme to break down that sugar, lactose, the disaccharide sugar, lactose. So if you're missing that, some lactose intolerant adults or lactose intolerant people can drink things that that don't have lactose in it. All right. So now these enzymes, sucrase, maltase, lactase, they are used to break down disaccharides into monosaccharides, and yay, we get down to mono, meaning one saccharide sugar, one sugar. So we went from many sugars to two sugars to one sugar, and fortunately the monosaccharide is the sugar that we can now digest. Carbohydrates provides energy and structural support. Humans can only utilize monosaccharides. That's all we can do. Glucose, fructose, galactose. These are the three monosaccharides that we can digest. Glucose is the big star of the show. That's what you really want. Have you ever met anybody that was on a carb-free diet? You will know because they are evil. And why are they evil? Because their brain is not getting enough glucose. The brain requires glucose as its energy source. So you don't eat carbs. Oh, I'm off bread. No, I'm not going to have that apple. No, I'm not going to have that sugar. No, don't put sugar in my tea. Yep, you're going to end up evil and difficult to live with. So because your brain requires glucose, and if you're not eating any, you're not going to feel too great. Now cellulose, that's something that grazing animals benefit from. So say on the campus of Wayne County Community College, there's lots of grass. They can go out there and graze on that grass, and they can get really big, like horses and cows, etc. Because they have the enzyme cellulase, whereas humans lack that enzyme for the most part. So cellulose, even though it is a carbohydrate, cannot be digested by humans because they lack the enzyme cellulase, right? I will leave it at that, except for the point that I know from some other readings that I've done, is that some people indigenous to the island of Japan for centuries they've been eating a lot of seaweed, and they can now digest that seaweed and have developed an enzyme that will digest seaweed, like when you have sushi or something like that. So that is just something to keep in mind. Okay, let's look at lipids. Lipids are fats, and they're nonpolar. I want you to know that lipids are a superior source of energy. Actually, much more than carbohydrates. And of course, it does not dissolve in water. Lipids are composed of fatty acids and glycerol held by ester bonds. Lipids may be saturated with no double bond present. That would mean that they're solid at room temperature, like butter, for example, butter. And they could be unsaturated with at least one double bond, and that would make it a liquid at room temperature. That would be, for example, oil. Before I go on though, I want to make sure that you look in your text and make sure you go over table, I think it's 2.3, to look at the functional groups. So you'll know when I say something is by ester bond or disulfide bonds. I want you to know what that means, so make sure you look at the functional group table in chapter 2. I think it's table 2.30. Okay, so let's take a look at this. Let's take a look at this. So here are going to be, really when you make a lipid, it's really a lot of carbon atoms. So a lot of carbons here in a chain or a link like this, a lot of carbons. Carbon can have a maximum of 4 other molecules attached to it, a maximum of 4. So for carbon here, with no double bonds like this, these would be hydrogens sticking off of these things here. Hydrogens. And you can see this carbon has 1, 2, 3, 4, and I'll just put a hydrogen here and here and here, so you'll know that these bonds have hydrogen and carbon attached to them. Here's a carbon here, 1, 2, 3, 4, and just all of these ones that I don't have the hydrogens written on it, just know that those would have hydrogens on it. And so that makes this chain saturated, because they all have hydrogens connected all around them. On the other hand, the unsaturated one, and the saturated one is like butter or margarine. Margarine, I really don't know what margarine is. It's some chemical made up, but butter for sure. And we're supposed to limit our saturated fats. So the ones that are like solid at room temperature, we're supposed to limit those. The unsaturated ones that have at least a double bond. So let's see what carbon these look like. And I'll put 2 double bonds in here so that you can see. So these will have hydrogens, of course, attached to them. And hydrogen. But since this has a double bond here, 1, 2, 3, 4, it doesn't get anything here. So it's unsaturated. It's a space that's empty here. This carbon here, 1, 2, 3, let's just put 1 here. That one doesn't have anything up here. So nothing here. And then this carbon is 1, 2, 3, so we'll put one here, but they don't have any, it doesn't have anything up here. This is 1, 2, 3. We'll put a hydrogen up here, but it has nothing there. And of course, this one is saturated with. So these are unsaturated because you don't see hydrogens here. Okay, and this is what equate to something really liquidy at room temperature, such as like an oil, olive oil, or vegetable oil, etc. And that's the fats that we are supposed to be able to have a little more of, more generous. Let's look when we're talking about fats. Let's continue on about phospholipids. This is an important fat for our cells because the phospholipid, the cell membrane is composed of phospholipids. It has a phosphate group, which has polar heads, and that means they're hydrophilic. They like being in water, these polar heads. And then it has also a fatty acid group that is nonpolar, the nonpolar tails, which are hydrophobic. The tails don't want to be in water. So let's take a look and see what that will look like. So we have the things that the heads are in water. So I'm just going to draw these polar heads. And it's a bilayer, so it's going to be 2 layers. And I'll put the tails inside here like this. Actually, they're a lot closer together. They touch. And then here's the other layer, since it's a bilayer, meaning 2 layers. And here are the tails here. The heads are outside. So this will be water out here and water here. But no water in between because the tails are nonpolar, right? The heads don't mind being in water, but the tails do not want to be in water. So when we draw a cell, and I draw a cell like that, and I just say, okay, this is a cell. Here's the nucleus. Here are the ribosomes. Here is the Golgi body. Here's the endoplasmic reticulum. Here's the mitochondria, all that. What we're drawing when we make a circle like that, that's really this lipid bilayer. Instead of us taking all these and making a lipid bilayer, really they touch all the heads and touch real close. Instead of us taking time to draw a lipid bilayer, we just make a circle. So that is what all of our cells are like with this lipid bilayer. And it's important because it is very specific about what it lets into a cell and what it lets out of a cell. So what you let in through this and what you let out through this lipid bilayer is going to be monitored. So that lipid bilayer is essential to our well-being. So let's just say that fats is the source of energy. Fats are much higher energy than carbohydrates, and that would provide us with a lot more energy source, right? So with this one, the phospholipids, I wanted you to know that they contain 2 fatty acids, right? And it's attached to a glycerol, and the 3rd glycerol binding site holds a phosphate group. So, let's see. The phospholipid bilayer allows the characteristic nature of the membrane to be selectively permeable to fluids. Is there anything else I want to say about this phospholipid bilayer? No, not really. This is important, but the gist of it is so you understand which way the heads and the tails are, and it is a bilayer, it's 2 layers. So I want you to understand that part, and it is for a fat, a lipid. Let's look at proteins. Proteins are composed of units known as amino acids, which I will abbreviate as AA from now on. From there, the building blocks of proteins are amino acids. So proteins are made of amino acids. And they exist in 20 different naturally occurring forms. 20 different naturally occurring forms. I'm not going to get into all those. But the peptide is composed of a short chain of amino acids. Polypeptide, you can see that's going to be a lot of peptides. It contains an unspecified number of amino acids, usually more than 20. So you can have a lot of amino acids, more than 20. Let's look at their structure. The structure is going to be vital. So a primary structure is a linear amino acid sequence that looks like this. It's just a linear, just amino acids all lined up together in a sequence. Secondary structure is a linear amino acid sequence with folds to form a helical structure of beta pleated sheets and alpha helices, right? So this will be more like, let's see, the drawing is going to be kind of funky, and then something like that. So you have the alpha helix here, alpha helix, this is the secondary structure, and then the beta pleated sheet here. Okay? Along with a little bit of the linear sequence, linear amino acid sequence here. So when you get to secondary, please know that that is when the beta pleated sheet and the alpha helix comes into play, right? Tertiary structure is a secondary protein, which is this one, with the alpha helix and beta pleated sheet, folded onto each other. And the main thing you need to know about tertiary contains three things: the beta pleated sheet, the alpha helix, and now it's got disulfide bonds in it. Disulfide bonds. So if I could try to draw it, I will put the sulfide bond. You see there's a double bond there, double bond right here. And I think that's with a cysteine. You'll have to look that up in the table 2.3. I want you to go over that 2.3 table and see if that's cysteine. The reason, so this is a tertiary structure, a little more complicated and has the disulfide bonds in it. Protein, okay, so let's just think about it. Protein. You know, your hair is protein. It's not alive. It's actually excess protein. It's dead. People that have curlier hair have a tendency to have more disulfide bonds. So their hair is got more tertiary structure in it. It's curlier. It's more coarse and it's curlier. But if that person wanted to say, I kind of want, I don't want my hair so curly, I want to straighten it. Well, you can denature the protein by adding heat or chemicals, like acids, or what we call relaxers or something like that. That will break these disulfide bonds and make the hair more relaxed and not so curly. You can add heat to it, which a lot of black women straighten their hair when they don't want it to be in this naturally curly state with the disulfide bonds. They'll add heat to it and then it can be straightened, and they can wear it different ways. There's a lot of things to do with that. So that is one thing to do with protein. And then quaternary structure. Quaternary structure is a secondary and tertiary structures. These two, beta pleated sheet, alpha helix, the cysteine, the disulfide bond, the beta. So it's the secondary and tertiary structures will come together to form functional groups such as enzymes and hemoglobin. These will be quaternary, etc. So hemoglobin, just remember, is a quaternary structure. And that's really combining the secondary and tertiary structures. Enzymes, of course, we talked about enzymes a minute ago. Enzymes are organic or biological catalysts that help speed up the rate of chemical reactions. We know that enzymes are specific due to their active sites. So, for example, you can have on a cell, you can have these things like this on the cell, and the enzymes will come around and they will land in here very specifically, and they will be shaped of the shape that the receptor is. That's why on cells, so you can see the enzymes in the dark blue, they are the shape of, here's the receptor, and here comes the enzyme. And it will fit directly in there, because you know enzymes and substrates are specific. So that's why you can have a lot of cells, and it seems like all the cells know exactly what to do, and that's because they have different receptors on them that can only accept the specific enzyme that is already in our bodies. So that helps explain, that helps explain the whole chemistry of enzymes and receptors. Okay, let's wrap up this chapter, ending with nucleic acids. So nucleic acid, this is the basis for genetic or hereditary function. This nucleic acids are what make every living thing what it is specifically, right? It's composed of monomers known as nucleotides. Adenine, A will stand for adenine, G will be for guanine, C for cytosine, T for thymine, and U for uracil. Each nucleotide, so each one of these, is made up of three components. You have a nitrogen base, the A, T, C, G. Then you have a pentose, which is a 5-carbon sugar. Pent means 5-carbon sugar. Then you have a phosphate group. So you got the nucleotide, the sugar, the pentose, and a phosphate group. Deoxyribonucleic acid, D, N, so you got the D, the N, and the A, that makes DNA. The pentose sugar is a deoxyribose, that's hence the name deoxyribonucleic acid. It exists as a 3D arrangement known as a double helix. It's kind of weird to draw, but it kind of looks like if you could think of a spiral staircase like that. The nucleotides for DNA are A, G, T, and C. Notice there's no U, no uracil for the DNA. It's a double-stranded superhelical molecule. The strands are mirror images of each other, so this strand here will look like this strand here. They're mirror images of each other, and they're specifically paired. In other words, an adenine can only be paired with a thymine. Guanine can only be paired with a cytosine. An A goes with a T goes with an A G goes with a C, and a C goes with a G. That's the only way they're paired. Otherwise, if one of these goes with another one, A goes with G or T goes with C, that's going to cause a mutation. So the DNA contains the genes and forms the chromosomes in the cells. This is what DNA does. It makes you, the person, uniquely of who you are. Genes expression occurs, of course, during division. So gene expression occurs during division. Ribonucleic acid, RNA, on the other hand, is the pentose sugar is a ribose. It's a single-stranded molecule. So instead of it being a double helix like this one, it's just like that. And it's used to express the DNA code into a protein. Uracil is used instead of thymine. So instead of the A being attached to thymine, T is attached to uracil in the RNA. The G is still accompanied by the C, and the C and the G go together. Please note there's no thymine in RNA, no uracil in DNA. I want to bring a point up that two guys got credited, Watson and Crick, for discovering and breaking the code of the genetic code called DNA, but truly it was a woman. Rosalind Franklin was the woman who really broke this and said, oh, this is a double helix. It is, it's a double-stranded superhelical molecule. She didn't get credit for it, but this is why I tell you to write everything down and do not use whiteout. Just draw a line through it if you think it's a mistake. Because her lab assistant was able to, after of course these guys, Watson and Crick, got the Nobel Prize in science, he was able to prove to them through her notebook, through her notes, that she was the one who actually came up with the discovery of the shape and structure of DNA. And she went through so many rounds of radiation to figure this out that she died at a young age. She was in her 30s from so much radiation. I think she ended up with some kind of cancer. I want to say ovarian cancer, but I can't remember exactly. But she ended up with cancer due to being in front of the machine, getting radiated so much, trying to figure this out. But much to her credit, now she has schools named after her, and she is being recognized. So many women in science have had to succumb to males taking their credit. So I just want to give a shout out to Rosalind Franklin. Adenosine triphosphate, of course, is ATP. So you can see the A, the T, and the P for ATP. It contains a nucleotide adenosine, and that's going to be involved in the transfer and storage of energy in our cells, right? ATP is our energy. So here's the phosphate, 1, 2, 3. That makes it a triphosphate, right? So that's triphosphate, and it's adenosine, so it's an ATP. As you lose energy, for example, like this is a high energy state. This is the highest energy state we have, ATP. Say you're sick and you start to deteriorate. You start to lose energy. This goes from a triple phosphate to, let's take one off, to a diphosphate, let's take another one off, to a monophosphate. And this is when they call in the family and say, I think you need to start telling your loved one goodbye. The heart rate starts to go low, the person doesn't eat or drink anymore, they're not talking, and you know that energy state has gotten lower and lower. So they call the family in. A genotype is a genetic makeup of an organism. The 23 chromosomes that you got from your mom and the other 23 chromosomes that you got from your dad make you a 46 chromosome human. That's your genotype. Those are the genes that you inherited. A phenotype, on the other hand, this is a protein product, or it is the outward appearance of an organism. Like, we don't know how those genes are going to come together to make you look, but how you look is your phenotype. What you're made of is your genotype. So you can actually have, for example, we see this a lot, like in the black community, the person has a genotype of a white parent and a black parent, but the phenotype, the way they look, is typically, they look what our society now labels as black. So, but the person is just as much genetically, they're just as much white as they are black. But how do they look? They generally are gonna look black, so that's a phenotype. You could just take Barack Obama, for example, if you just wanted to have an idea about that. His genes are just as much black, he's half black, half white, but phenotypically, he looks like what society has deemed a black man. So this whole thing about what you are is totally political, has nothing to do with science, it's not based in science. Race is not a science-based fact. So keep that in mind as you go forward in your life, and try to just stick with the science of things. All right, so that actually wraps up chapter 2. We're going to end it there, and we'll get into chapter 3 next time.